introduce the next session, where we will hear from IMS and NASH bursary winners. So the first speaker is Dr. Elise. She's postdoctoral research fellow working with Prof. Kui Yong at UCL. Her main work has been on NK cell compartment, and she'll share with us her findings. She is sponsored jointly by UKMS as well as Binding Site. Thank you.
These are not my slides. Okay, these ones are. So I'd just like to start by thanking the organisers for giving me the opportunity to present some of this work here today. I'm a postdoc working in Kui Yong's lab at UCL, and my work focuses on characterising the NK cell, or the natural killer cell compartment, in multiple myeloma, and specifically how this changes with disease progression. We all know that multiple myeloma is preceded by clinically defined precursor states, namely MGUS and smouldering. We know that progression through these states is accompanied by increasing tumour burden, the acquisition of secondary or additional genetic events, and ultimately an increasing risk of developing overt multiple myeloma. We also now know that the plasma cells interact with their bone marrow or microenvironment, but what we don't understand is what impact this microenvironment may have on tumour progression. This is why our lab has set up the COSMOS study. Some of you may have heard of this already. COSMOS is a UK observational study recruiting patients with precursor disease, namely patients with smouldering myeloma. And the idea is to track both clinical and biological samples from these patients through the course of the trial, or unfortunately up until the point the patient progresses. And the overall aim of COSMOS is to identify both clinical and biological features that are associated with progression. And using these samples, we're able to investigate a number of features, including different immune cells that may contribute to this progression. My particular interest and the focus of this talk is the role of natural killer cells. And the overall aims of my work has been to characterize the NK cell compartment within the bone marrow, which is fairly not well understood, and how this compartment is altered through the progression of multiple myeloma. This includes looking at the functional capacity of patient NK cells. And the overall aim really is to explore whether NK cells have any role in the progression of multiple myeloma from precursor state. But for today, I'll just be focusing on the first main aim, which is to characterize what NK cells are actually in the bone marrow. So each bone marrow that we process within the lab at UCL has a basic flow cytometry panel run on freshly isolated MNCs. This panel includes markers to identify tumour, so CD138 and CD38, but also a number of markers to identify key immune cell subsets, including those for NK cells. And based on the relative expression of CD56, we can identify the 56 bright, and these are typically considered the more immature subset, cytokine producers. We can also identify the 56 dim, and these are the more cytotoxic subset that also express the Fc receptor CD16, enabling them to perform ADCC through monoclonal antibodies such as Dara and isatuximab. When we looked at the overall abundance of NK cell proportion in the marrow in our patients' cohort, we found that there was no overall difference in the overall frequency of NK cells. However, there was a stepwise increase in the proportion of those NK cells that had a 56 bright or more immature phenotype. The abundance of this 56 bright immature phenotype was associated with tumour burden in both precursor disease and those patients with multiple myeloma. The relationship between tumour and the abundance of these 56 bright cells was further elucidated through the longitudinal sampling that we have of our COSMOS cohort. So here we found that fluctuations in the tumour burden, which is in the blue here, was reflected in changes in the abundance of the 56 bright NK cells, i.e. if tumour goes up, so did the abundance of these 56 bright cells. And these results highlight that there's a dynamic relationship between tumour burden and the skewing of the NK cell compartment in the bone marrow. To gain granularity in terms of phenotyping the NK cells in the bone marrow, we employed single-cell RNA sequencing. So this was done through the help of Isa, Kane, and Louise in the lab. And this data set includes patients from our Cosmos cohort, which was integrated with publicly available data. This data set includes patients from MGUS, smoldering, and myeloma, as well as those with from healthy donors. The data set comprises of over 100,000 NK cells. This is one of the largest NK cell data sets to date, and certainly the largest of bone marrow NK cells. And from this, we were able to identify the main NK cell subtypes, but most interestingly for us was the identification of a bone marrow specific population that is absent from the blood. Further characterization of this subset revealed that this bone marrow NK cell population shared characteristics to the 56 bright with high expression of granzyme K and chemokines such as XCL1 and XCL2. However, this population was also found to express high levels of tissue residency markers, CD69 and CXCR6, confirming that this is a bone marrow tissue resident population. We used this data set to gain insight into the kind of functional role that these cells may be playing. And we found, when compared to the 56 dim and the 56 bright, that the bone marrow resident cells were less enriched for genes associated with cytotoxicity and cell killing. However, were more enriched for genes associated with inflammation, chemotaxis, and cytokine production, suggesting that these NK cells, whilst not cytotoxic, may have a role in inflammation or immunomodulatory properties. Looking across our patient cohort, we found that it was actually this population that increased from MGUS to multiple myeloma. Again, because we have the Cosmos longitudinal data, we're able to track these changes in our own patient cohorts. And this time, changes in the tumor burden, again in blue, was mirrored by increasing proportion of these bone marrow tissue resident cells. We confirmed by flow cytometry within our own patient cohort the presence of these bone marrow tissue resident cells by co-expression of CD69 and CXCR6, and we find that they are present in the bone marrow but not in matched peripheral blood. In a small cohort of our own patients, we found that on average, myeloma patients had more of these tissue resident cells when compared to precursor. So in summary, we find that myeloma progression is associated with a skewing of the NK cell compartment towards a more cytokine-producing or more immature subset. And we find that this skewing is associated with tumor burden. We've utilized single-cell RNA sequencing to generate one of the largest data sets of NK cells. And this was used to identify a previously unidentified bone marrow tissue resident NK cell subset, which we believe may be involved in the inflammation of the marrow. If anybody's at all interested, we have, this is now on bioRxiv. I just want to finish by thanking the UCL Myeloma Lab, CRUK for funding this work, all the patients that have kindly donated their samples to either Cosmos or Radar, and finally to the UKMS and Binding Side for awarding me the Travel Award to present this work in Rio last year. Thank you.
Are there any questions?
Thank you very much, Elise. It's very, very nice work. Did you look at the spatial analysis? I mean, did they look like they are actually interacting with the myeloma cells? Or did you do an interaction analysis on your single cell data set?
No, that's a good question, and that's something I'd like to do next, is actually look at where these bone marrow resident cells lie in the bone marrow, and specifically proximity to tumor and possibly other immune cell types. Yeah, something in progress.
Thank you very much. Thank you.